Okay, class, welcome to chapter 22, section 4 on DNA. So we're talking about different genes are expressed differently in different cells. So expressed, remember we have the exons and the introns. The introns are the ones that are taken out. The exons are spliced together and expressed. So all of the cells have all the DNA in their nucleus, as long as the cell has a nucleus, but only some are expressed. That's how we get different cells and then thus different tissues. Remember, neck, muscles, nervous, epithelial, connective is because different genes are expressed. So we have different gene expression regulation, so we're regulating that. One way is pre-transcriptional control. So that's even before the DNA makes the RNA. So access to the DNA in the nucleus can be controlled because remember the chromosomes are usually condensed. So in order to decondense the chromosome, we can have pre-transcriptional control by just not decondensing part of the chromosome or decondensing the chromosome so that the DNA can be stretched out and make the mRNA. Then there's transcriptional control, so that's when the DNA is making the RNA. We have factors that must initiate transcription, and then there's initiation of translation. So the mRNA binds to the smaller of the two ribosomal subunits. So we already looked at that. Then we have post-translational control. That's just how the mRNA is processed in the nucleus with the introns and the exons, and how fast it breaks down in the cytoplasm. So in some cases we can mark the mRNA for destruction by proteases, and then it's not in there as much, it's not making as much protein. So there are a lot of things we can do. Okay, DNA sequencing is how we learn about DNA, and it takes several hours to sequence 1,000 base pairs of DNA sections. And how do we do that? We use PCR, which stands for polypolymerase chain reaction. And to do that, we take Taq polymerase, which we borrowed from an archaea, an extreme archaea, and because of that, we can make many copies of the DNA to be sequenced. So we can use this in crime. We can analyze the unique repeat patterns of individuals, and that's called DNA fingerprinting. Also, we can, with modern day sequencing, we can detect dyes in the nucleotides with lasers, which is so much easier than electrophoresis, believe me. So we can also use recombinant DNA technology. I talked about this a little bit, and I'll talk about it more in class. This is how we can make insulin for human health. So how do we do that? We will do some cloning. So with cloning, we need a host cell, such as a bacteria, a restriction enzyme, which will cut the plasmid so we can insert the gene for human insulin, then a vector, which will carry the plasmid into the bacteria. So we do that, we can make insulin for human health, and the people who write the test want you to know that you also do this for frost minus substances for plant health. So plant health, we get all our food from plants, so it's linked to human health. Okay, so we decided that we would, since it only took several hours to sequence 1,000 base pairs, it used to take longer, that we would decode the entire human genome. So it took many, many years. I remember in 1999 we had neighbors in our apartment who were actually working on it. The lady was from Turkey and she was working on it. They were very nice neighbors, by the way. But anyway, it was completed in 2003. You need to know that. And we found that our genome is 85% similar to a mouse. So I drew a mouse here. And because of that, we're able to use mouse models to study cystic fibrosis and other diseases too, but cystic fibrosis is the one you need to know. And then we have something called functional genomics. The goal of functional genomics is to determine the purpose of our genes and how they work together to create a human being. And a lot of these guys actually look at other species to kind of figure this out. So these researchers, they found 3 genes on chromosome 22 on chimpanzees, believe it or not, for insight on speech development, hearing, and smell genes. I don't know what that has to do with insight into our evolution. I have no idea. But anyway, that's what you need to know, specifically speech development, talking, hearing, and smell with the chimpanzee. So that is let's see, I think that is everything. I did want to mention though that when the researchers first did the Human Genome Project, when they first started, they expected they would find 80,000 genes in the human genome, and actually they only found 19,000, 20,000 according to your book, but 19,000, remember the most important guy is the guy that writes the test, 19,000 according to him, and only 2% of the human genome is used to code for functional proteins. So what is the other 98% used for? I don't know, maybe recording. Okay, so I will do one more short segment and then I will see you Monday.